right, folks, for this final video, we're going to talk through the nature and nurture issue. And we've already touched on this a little bit when we talked about heritability, right? So ultimately, rather than saying, is this psychological trait due to nature or nurture, I think most psychology students at this point understand that basically every trait is going to involve some nature and some nurture, some genes and some environment. When we talked about heritability, we learned that heritability is typical for a lot of psychological traits, personality traits, IQ, even susceptibility to psychological disorders, substance use disorders, that sort of thing, runs in families, right? But it's incomplete, right? So identical twins aren't exactly the same. And when they're raised separately, they're a little bit different, right? So heritability is typical, but it's not 100%. So there's some nature, some nurture. So the real question is going to be, how do we work with genes and environment? How do those genes interact with the environment to lead to the development of a complex human being with complex human psychology, right? So that's really the nature and nurture question, is not which one, but um, partly how much of this trait is heritable, which we talked about, and then how do nature and nurture interact? So really the first question is, how do genes do anything, right? How do we go from these simple DNA sequences, which are just chemicals, adenine, cytosine, thymine, and guanine. You don't need to memorize those. Just built into different orders. It's just those four on the DNA strands anyway. That are built into these DNA sequences. So, how do those serve as building blocks to lead to all the complex psychology that we have, or at least to lead to the inheritance of those psychological traits? Well, every nucleus in your cells, starting with that single zygote that we get, has DNA sequences and genes in it, and those are going to undergo what's called gene expression. So you have the genes. The genes don't do anything by themselves, but then they get expressed, and that's going to lead to development. And it leads to a bunch of other stuff too, right? But you may have heard about gene expression. You may be familiar with the terms transcription and translation. So you'll see those again here. There's four steps to it. I list them here, and then there's a figure on the next page. So let's walk through it twice. Number one, that DNA strand unravels. You see they're bound, these double helix set up. That double helix unbinds itself so that there's something to attach to. And then we have this stuff called messenger RNA or mRNA. You might be familiar with mRNA from vaccine technology, right? So that's not DNA, it's RNA, which can come in and read. Um, Transcribe, copy, right? The DNA, okay, the mRNA comes in and is synthesized. It's called transcription. It's called just transcribing, copying a piece off in mRNA form, and then it leaves the nucleus. So it heads out of the nucleus of the cell. The next part is the reading. So we've got the transcribing, the copying, and then the reading in a kind of new language. And that happens in the ribosome. So the ribosome of your nucleus reads the mRNA in these sequences of three, and that creates amino acids. That process is called translation. So generally, what I want you to know is DNA unravels, mRNA is transcribed, and then mRNA creates amino acids, translates into amino acids. Okay. Finally, the amino acids, you may be aware, are building blocks, right? So amino acids build up to become proteins. Protein then become more complex structures. And so the brains that we have that are going to do a lot of the psychology that we're going to talk about, the brains are going to be developed from building blocks that ultimately started as amino acids, and those amino acids were translated based on genes. So that's going to be the basic mechanism from how we get from genes, and then depending on where we are in the body, those genes are going to lead to development of different structures. Here's a figure form of that. It's in your textbook as well, so go ahead and review this if you want to spend some time on it. But here's the four steps again. So we've got the unraveling, the transcribing. The mRNA is transcribed from the exposed DNA. The mRNA itself doesn't do anything, right? But the ribosome comes in and it translates that mRNA and then creates amino acids, which then can be completed into proteins. So that's it. It's a basic mechanism, right? We've got this DNA sequence that can lead to the development of different proteins. And given that my DNA is a little bit different than your DNA, I'm going to get slightly different proteins. And so my brain's going to look a little different than yours. My eyes are going to look a little different than yours, right? All of those are going to be based a bit on the genes that start the whole process of a gene expression. And the other piece to this is that gene expression doesn't always just go in a certain way, right? I noted that depending on where you are in the body, different genes will lead to different things, right? And it's also true that at different times in your life, different genes will get expressed. And so there's a regulation process of this. Our body has a regulation mechanism to determine which proteins to produce at which rates and when and where to do so. And so your and my genes might be a little bit different, but our regulation mechanisms might be a little bit different. And those regulation mechanisms are started by genes too, right? Um, for the most part. So, as examples of gene regulation, we've got things that are enhancers. So, these will initiate the synthesis of proteins or increase the rate of synthesis. So, you've got a sequence of DNA that doesn't itself get transcribed, but it will essentially turn on the transcription of the next sequence or increase the rate of transcription of the next sequence. So, you and I might have the same sequence here. But we might have different enhancers. And so I might get more of this or less of this than you do. Transcription factors bind to DNA and influence. And so these aren't on the DNA itself. They're factors that bind on top of it. So they'd be produced and then bind on. And so they might do that, in some places and not others, or at some times and not others. And so a gene might not get expressed unless it has a transcription factor attached to it. There's a general term called epigenetics that refers to the pattern of actual gene expression versus the genes possessed. And so there's a number of factors, transcription factors, and a few other examples we'll look at on the next page that are not in your genes themselves, but that can affect gene expression. And these are called epigenetic factors. There's an interesting whole area of research about how some epigenetic patterns can be heritable too. So I can inherit actual gene sequences from the chromosomes of my mother and father, but I can also inherit epigenetic factors. So my epigenetic factors could come from my father, and some of those epigenetic factors can be affected by experience and that sort of thing. So gene expression is not always just on a formula that's always going to work the same way. It's going to be expressed in different ways based on enhancers, transcription factors, and other epigenetic factors that's going to influence gene expression. There's a video in your lecture slides that breaks this down a little bit more in terms of epigenetic mechanisms. Um, and it'll talk about two additional examples. I generally want you to know the general principle of epigenetic factors and not necessarily know how every one of them works, but just to give you a flavor of two more versions of this so you can understand the general idea. Excuse me. So one of them is called histone remodeling. So in this case, chromosomes are actually wrapped around these other chemicals called histones. And if the histone changes shape, that's going to affect whether the DNA is transcribed or not based on where we are in this shape, right? If it's nice and smooth around the outside, it'll get transcribed in a certain way. If it's going around a corner, it might or might not get transcribed around that corner. So just the shape of the thing it's surrounding can affect whether DNA is expressed or not, right? That's not the change to my genes, right? And so it's not genetic. It's beyond that. So it is epigenetic. That's what epi means. It means above or beyond genetic. Okay. So if my histones change shape through experience, or if I inherit different types of histones, right, that can change how my genes are expressed without changing my genes. Another epigenetic factor is DNA methylation, where a methyl group can attach on. Okay, this is like a transcription factor, right? It attaches on here, and it's going to determine whether the DNA gets expressed at this base pair, right? And so, again, the DNA is not getting changed. The DNA is the same, right? But if I get a methyl group added and you don't, then we'll have maybe the same DNA sequence, either expressed or not, at different rates. So this is epigenetics. And epigenetics is a nice example of how genes are going to have to develop over time in an environment that's changing, right? And so histones can change, methyl groups can get added, other transcription factors, there's a whole bunch of them, right? And that can change with experience over time. So the same genes can express differently over time and with experience. And that's what we're saying here, right? So, gene expression and epigenetic mechanisms may be able to count for or predict gene experience interactions. And so, um, and so we know that we have genes and we have experience. Um, but we want to think about them as interacting rather than just adding together, right? So you don't just have like 10 points of experience and 10 points of genes, and that's going to lead to a certain amount of extroversion, right? These things are going to interact with each other. So experience can change epigenetic mechanisms, which can change whether genes get expressed, right? So depending on my genes and depending on my experience, they can work together or they can interact in different ways. There's a few examples um, of how this works sort of in real life and not at a chemical level. And so you can see that genes and environment interact. There's a couple examples we're going to walk through, and then your book talks about birdsong if you want to look for a third example. So the first example is selective breeding. And in selective breeding, you're changing genes through selection, right? Through artificial selection. This is like when you breed different dogs to be bigger or smaller or more violent or more cuddly, right? And in this case, we've got a classic research study. Um, from the 1930s, right? Where they were training rats to run in mazes and they selectively bred them to either be maze bright or maze dull. Right. And this means that they either made few errors in the maze or a lot of errors in the maze. So they put the rat in the maze. I say mice earlier. They put the rat in the maze and they counted the number of errors it makes before it gets to the prize at the end, right? And if it makes a bunch of errors consistently, then they called it maze dull, right? And if it made a few errors consistently, they called it maze bright. And in the original population, some of those rats had a hard time, some of them did great, and most of them were kind of in the middle, right? But there's a wide variety of rats. Cool. They then bred them out. So the Maze Doll went with Maze Doll, the Maze Bright went with Maze Bright, and they started to pull away from each other over generations, over 4 generations. We now have most of them are either bad at the maze or good at the maze. And then by the 8th generation, they're separated in terms of whether they're really bad at the maze or really good at the maze. And this isn't based on training. None of them are getting training like from their dumb parents, right? These rats are put in the maze, and presumably because of the genetics that they've inherited from their originally maze doll or maze bright parents over generation and generation and generation, these rats are just a little bit better at learning the maze for whatever reason. Then there's different explanations for why, but they just were better. Okay. So there's genetic inheritance, presumably. And so running a maze is based on genes, right? Well, research came around, and this is a figure from a more recent study, but research came around in the 1950s that suggested that this genetic inheritance isn't the only piece of the puzzle, right? So they put rats in either a standard housing environment like this or an enriched environment like this with lots of toys and more rats and more space, right? So we're affecting the environment here, right? So either the environment is enriched or standard. And so you might think, well, putting somebody in an enriched environment is going to be good and this is going to be bad. That makes sense. But they found an interesting pattern where it wasn't just, again, um, genetics plus environment. These things interacted. So what they found was that in an impoverished environment, in a standard kind of cage environment like this, they found the genetic difference that they were expecting, that they had found before. So the maze dull rats made more errors than the maze bright rats. Genes are controlling things here. So genes are important. Okay. Here, it wasn't that all of the rats got better in the enriched environment. What happened was that the genetic influence was reduced. So the rats who had sort of the genetic background that might make it harder for them to learn the maze actually caught up basically with the maze bright rats. And so the genetic influence was reduced in the enriched environment. Okay. So in this case, it's not that the genes don't matter anymore, but they matter depending on the environment, right? So they matter in this environment and they matter less in this environment. So both the genes and the environment are critical, but they interact. Here's another example of that, of how genes and environment interact. They don't just add together. And that is PKU, phenylketonuria. This is a condition that gets tested in genetic screenings, and it's an absence of a single gene. The single gene produces this chemical called phenylalanine hydroxylase. I'm not going to test you on that if you're in my class, right? Hydroxylase means that it's going to break down phenylalanine. Okay, so you got phenylalanine, and then you've got phenylalanine hydroxylase breaks it down. Okay. Now, most of us are going to make that, and it's going to break down phenylalanine. This is like most of us are going to have the chemical that's going to break down the enzyme that breaks down lactose, so we can drink milk, but not everybody does, so they get lactose intolerant. Same thing here. Most of us are going to be able to break down phenylalanine, but some people aren't because they don't have that gene or it doesn't develop in the same way. Now, the problem is if you get phenylalanine building up over time, that leads to developmental problems and intellectual disabilities. So, for a while, a lot of people with intellectual disabilities developed those intellectual differences because they had PKU and they were eating food with phenylalanine and it was affecting their brain development. Now, here's the thing. That only matters if you eat food with phenylalanine, right? So only when you interact with an environment that contains phenylalanine does this genetic difference relate to psychology, right? So we've got a genetic difference, a single gene that leads to pretty intense intellectual differences, developmental differences, one gene, right? So genes affecting intelligence, right? But if I change my diet, then the gene doesn't affect intelligence anymore at all. Right? So it's not the gene that's affecting intelligence. The gene is affecting how we interact with our environment and whether we can break down a chemical, and the chemical then leads to changes in development. So these are working in interaction with one another. Okay. Now the diet, if you got PKU, can be difficult, and I know there's lots of people working on different versions of diets and that sort of thing for this. But just as a basic understanding, talk to a doctor or dietitian if you want more details. As a basic understanding, there's lots of things with phenylalanine in them. So um, that's why they do genetic screening for it. So not so that they can tell you that your kid's going to have intellectual disabilities, but that they can start a diet and then you can develop normally. That's it for our end of chapter two on gene environment interactions or nature nurture issue. The most important thing I want you to keep in mind with nature nurture is that um, basically every psychological trait is going to involve some heritability to it, right? That's the genes and epigenetic inheritance and in interaction with the environment. Okay, learning, training, development, interactions, and that sort of thing. So it's both nature and nurture, but also they interact. So um, genes may or may not influence psychology depending on the environment. So they may influence psychology in this environment, but not that environment. Okay, so think about it that way, and you'll be good to go as we look at these interactions going forward.